yeah, you're going to receive a notification. Oh, yeah. Perfect. Yeah, thank you so much for doing this, Karo. I also learned that the time is a little bit later in your region.
Yeah, we're about 5 p.m. here.
Oh, okay, wow.
Artem?
Yes.
Sorry. Not used to it. Do you want some wine and shit?
But it's quite difficult to hear you from that distance. So maybe it might be a good idea in the question period that you come a little bit closer.
Yes, and I
Okay.
can also repeat the questions.
We will repeat the questions.
Okay.
And by the way, is it okay if I present you with what you sent me for the bio, or do you want me to add something else?
Oh, that's fine.
Okay.
Yeah, what I sent is great.
Okay, I will take my phone and yeah, we will start in 5 minutes.
Okay.
We will just wait. Usually people come really at, like on time, not before. So.
Oh, because
Yeah.
the students that use us or we.
Okay, nice. Yeah, we should discuss also, if you have time, the experiments.
Yeah. How's your experiment going? Noemi?
It's okay. It's working
Yeah.
actually. So we
I
are.
saw the paper that you sent on high energy cosmic rays.
Oh yes, the highest energy cosmic rays that we detected.
Yeah.
Yeah.
So those high energy cosmic rays are coming from our galaxy
No. Yeah.
outside.
So these very, very high energetic ones are coming from unknown sources, right? This is a mystery still.
Mhm.
This is what, like, we are trying to understand what are the sources. And because they are deflected by the galactic magnetic field, then we need a good model for the galactic magnetic field to be able to calculate the trajectories and see where it would point, right? Because we can detect it coming from this direction. And in reality, the source is there because it was deflected by the magnetic field on its way. So there are like candidates, we think maybe relativistic jets, like associated with black holes, supermassive or stellar mass black holes, or very, very large scale accretion shocks. But we still don't know. So this is what I'm working on to try to investigate the origin of these particles.
Yeah, and they are providing you with the muons, no, for your experiment, polarized muons.
Oh, so that are two things. There is the JPAC facility, which is in Japan. So for that, I met two researchers, Takahashi and Kobayashi, and they are irradiating amino acids with spin polarized muons using the JPAC facility. And then I also met with two researchers from the Origins Institute in Munich. And I actually tried to get to Munich last week, but it was impossible because of the snowstorm. And they want to actually irradiate DNA with spin polarized muons. So I'm very excited about that. At the PSI, so the Paul Scherrer Institute. Where you've been, yes, yeah, this is what I was. Hey, hi.
Oh.
How are you?
Have you been to visit?
Yes, so this is Karo, Michaelian.
David Dimmer.
David Dimmer. This is David Dimmer.
David.
Yes,
Yeah,
you can.
very well.
Yeah, Dave, you should speak to here because I'm not sure he can hear you.
Yeah, I'm sure he's gonna have a lot of criticism.
Yes.
Oh, I don't know about that. I just enjoy these talks.
Yeah. Well, I hope you enjoy this one, and I hope that I convince you.
Good. Where are you now? Where are you right now?
Oh, where am I? I'm in a little town which is called Temisco, which is about 100 kilometers south of Mexico City.
No kidding. How about that?
Yeah, so we're 2 hours advanced here. the sun's going down. You don't see me too well, but the sun will soon go down.
Yep.
And you get a better picture.
Good. Yeah, I've been to Mexico City several times. My colleague Antonio Lascano, do you know him?
Oh, yes, of course, Antonio.
I thought you would.
Yeah,
Well, I'm gonna get a bit of coffee before the talk starts. Excuse me.
sure. Hello, Julian.
Hi, Karo.
Thanks for coming on.
Happy to do so. Very curious. Thanks for the invitation.
You still remember the work that you did in your master's?
In the masters, yes. Hardly, but yes. Yes, I can still remember something.
Where are you now?
In Cuernavaca, we are really close.
Oh, really? Wow, you should come and visit then.
Sure, why not?
Are you staying here for some time or just visiting?
No, it's
Yeah, no, I'm doing a postdoc here in the Biotechnology Institute. So probably I will stay 1 year.
just one person. Yes.
Are you're doing your postdoc in the Institute for Investigation in Energy or something like that?
You know, in the Biotechnology Institute, I'm doing super resolution microscopy.
I didn't catch the
You can read
name
the
of
name.
the institute.
Thank you.
Biotechnology is
Biotechnology.
yes.
Is it associated with the university or what?
Yeah. It's from the UNAM.
No. Okay.
Yeah.
Biotechnology. I didn't know there was an institute here. Is it in
Yeah.
Cuernavaca or
Just in front of Ciencias Físicas.
okay, in La UAM?
Exactly, yeah.
Okay. Oh, great. I didn't know that.
Yeah.
How
I
long
will
have
invite you someday.
How long have you been in Cuernavaca?
Yes. In February will be 1 year.
1 year. Okay.
Yeah.
Oh, too bad you didn't look me up until now, but we'll get
Yeah.
together.
Busy, but we need to catch up. We
Yes.
definitely will need to do that.
Yeah, that's great. Julian, we have other students here. Andres, he's a student. How are you doing, Andres? He's a student of mine in a bachelor's degree.
Oh, nice.
I'm having trouble remembering my English.
It's normal. Okay, I let you attend your talk and we catch up later.
Okay.
Good luck and really curious about your
Yeah,
recent
thanks,
job.
Julian. Yeah, you send me an email.
Sure, totally. I will do.
Okay. Hello, Andres.
Hi, Karo.
How are you doing?
Yeah, I think I should.
Fine.
Okay. Are you gonna send me
Because I don't.
the latest version of your thesis?
Yes, I will soon send you the corrections about chapter 1 and 2.
Okay. Yeah, at least 1, 2, and 3. I think you have finished also, no?
You have the ability
Oh,
to.
yeah. 3, I already did the corrections you sent me last time, but I'm still working on chapter 2.
Okay.
Talking right now, so maybe not. But we will start, Caro, in 1 minute, okay?
Okay.
Just fine. You're probably busy as usual.
There will be more people.
Yeah, they got over that auto accident. That's good stuff. Once in a lifetime is enough. Yeah. That's right. Yeah, all my kids from my previous family have had very otherwise.
Are you recording?
I've been the
Okay.
holdup till recently.
So, Caro, we are going to start. Are you?
Crazy deal.
Okay.
Where you feel like-
Because we don't see you anymore.
So I think of myself running off the road.
Yeah, we don't see on the camera.
Yeah,
Karo.
your camera did something.
I think it's out of focus.
Okay.
You can't see me?
Oh, now we can.
Yes, now.
Oh, yeah, I guess the trouble is that I have got light in the background here. Got a sunshine.
Okay,
better?
well. Yes. Alrighty, so let's start. So we are very happy today to have Karo Michaelian as our speaker for our monthly astrobiology colloquium. So Karo Michaelian is a Canadian researcher at the Institute of Physics at the National Autonomous University of Mexico. His bachelor is in geophysics in 1981 and his PhD in nuclear physics, proton-proton Bremsstrahlung in 1987. Then he did postdoctoral work on pion absorption in nuclei at the Paul Scherrer Institute in Switzerland. He now works on nanophysics and the non-equilibrium thermodynamics of life and evolution. He has developed a robust framework for the UVC dissipative structuring of the fundamental molecule of life and their evolution, which he calls the thermodynamic dissipation theory for the origin and evolution of life. And yes, this is what he's going to tell us about. So the stage is yours.
Oh, well, thank you very much, Noemi, for the invitation. So today, what I'm going to be treating is a fundamental question. Is there a reason for life? Now, if you ask a biologist, they will say, no, there's no reason, no purpose for life. It's just that nature operates the way it does. If it doesn't, it wouldn't survive and it wouldn't pass on its genes to the following generation. But in fact, I believe that the question is that the answer to that question is that there is, in fact, a reason for life. And that is a thermodynamic reason or thermodynamic function. And that is the dissipation of sunlight into heat. So today on the plants, on the leaves of the plants, there are pigments which absorb the near ultraviolet and the visible light, and they dissipate that light into heat. And that is entropy production or dissipation. And the pigments in the leaves are produced through a process which is known as dissipative structuring of pigments, which is using photochemical and chemical potentials to produce those pigments. But at the origin of life, we didn't have these biosynthetic pathways, these complex biosynthetic pathways to produce the pigments. But what we did have is ultraviolet light in the UVC, or the far ultraviolet. And this light, each photon in this light has enough energy to break and remake carbon covalent bonds. So what I'm going to try to convince you today is that the fundamental molecules of life, that is those that are in the three domains of life, the archaea, the bacteria, and the eukaryote, these fundamental molecules were really pigments in the UVC. And their function or their purpose was to dissipate this UVC light into heat. Now, animals came later, and they are really only a footnote in this story. They are really the catalysts which spread the nutrients, the seeds, and they pollinate, among other functions. So they are serving the plants in this fundamental thermodynamic process, which is dissipation of sunlight into heat, or entropy production. So it's in line with the second law of thermodynamics. So if we look at nature today, we find an abundance of trees and plants on land and cyanobacteria and diatoms on the ocean surface. You'd be hard-pressed to find an animal within any of these, the regions of these pictures. And we can quantify that. And in fact, that was done by Bar-On et al. In 2018. They made some estimates for the amount of plants, animals, and bacteria and other organisms which are in the biosphere. So plants and cyanobacteria make up by far the greatest amount, about 95% of all of the carbon biomass in the biosphere. So for example, plants make up about 450 gigatons. Each one of these green blocks on the left of the screen corresponds to a gigaton of carbon biomass. Animals make up only 2 gigatons, which are the small gray blocks at the bottom in the corner of your screen here. And humans, in fact, make up only 0.06 gigatons, which is this little red slab. And as I said, there are other organisms like bacteria, fungi, archaea, protists. But in total, the plants and cyanobacteria and the diatoms make up about 95% of all the carbon biomass in the biosphere. So what are all these plants and cyanobacteria doing? Well, they're absorbing free energy from the sunlight. And if one looks at the absorption spectrum of this of plants, this graph here is an example of 4 different plant leaves. They're absorbing very strongly, about 90% over the entire spectrum. Okay, here we have the wavelength in nanometers, but it's on a logarithmic scale. So here we go down to about 300 nanometers, 0.3 microns. But in fact, this graph goes all the way down to about 200 nanometers. So from about 200 to about 700 at the red edge, we're absorbing very strongly almost all of the light in the leaves of the plants. Now there's a little dip at about 550, and that corresponds to the green, and that's why we see the plants in the green. This light is not absorbed very well. But there is this sharp cutoff at about 700 nanometers, and then little absorption. And then again, we start absorbing at about 1.5 or 1 micron, which is due to the absorption in water in the leaves. So the leaves are absorbing a lot of the free energy, almost all of it. Now, what are the plants doing with this free energy? Well, the first answer might be photosynthesis. But in fact, if you look at the efficiency of photosynthesis, we find that only about 1% of the free energy is actually being used in the construction of carbon covalent bonding or photosynthesis. The rest, which is about 99%, is simply dissipated to heat. And sometimes the biologists call this non-photosynthetic quenching. So I should mention that the absorption in the leaves is due to a number of pigments. These are pigments which exist today. And they're finding new pigments every year. So there are many, many pigments in the leaves, not only the ones that I have listed here. Now, because of the absorption of the photons in the leaves and in the bodies of the cyanobacteria and diatoms on the surfaces of water bodies, we get heat deposited, and this heat increases the evaporation. So plants and cyanobacteria are coupling to the water cycle, and that is a further production of entropy because we are converting infrared light at the surface of Earth and then sending it out to space. When the water condenses and it releases its energy, and that's emitted into outer space at a wavelength which is very redshifted. So it's like 2 irreversible biotic. One is a biotic process and one is an abiotic process, but they're coupled together, and together they increase the rate of entropy production. So therefore, photons and pigments, which are in life, and water gives rise to dissipation. So we have the near ultraviolet and the visible light coming into the water and being absorbed, for example, on a chlorophyll pigment, and that being dissipated, that light, into infrared light. So life's thermodynamic function then is dissipation of photons. And this occurs in plants on land or in cyanobacteria and diatoms on the ocean surface. Now, if we look at a more global picture, then we see that the sun is emitting in a narrow beam its light, and it's impinging on Earth. Part of that is scattered, about 30%, which is the albedo. The rest is absorbed by the atmosphere and the Earth's surface and converted into infrared light, which is emitted into outer space. So what the biosphere is doing then is shifting the spectrum, the input spectrum, which is the spectrum here in blue at the bottom, shifting it from the near ultraviolet and visible into the infrared. So it's not only light, but also the abiotic processes. For example, like A is the water cycle, but it could also be winds and ocean currents. Now, if we calculate the entropy production, which is easy to do because we know from satellites what is the incident spectrum from the sun, and we can also measure with the same satellites the outgoing spectrum of Earth, and knowing that there's a relation between the spectra, and the entropy, so the entropy of an arbitrary beam of light, which was described by Planck in the early 1900s. So there's a very simple formula for that. So given a black body spectrum, or whatever spectrum, doesn't have to be black body, but given a spectrum, we can calculate the entropy of that light. And by knowing the flux of the light into our system and the flux out of our system, so this equation here at the bottom, we can calculate the entropy production of the biosphere. And if we do that, and if we compare it with our neighboring planets, Venus and Mars, we find that Earth is about twice as effective in producing entropy as either of the neighboring planets. So, well, what about at the origin of life? Well, at the origin of life, there didn't exist these complex biosynthetic pathways. So we didn't have any visible pigments. But as I mentioned, what we did have is UVC light and maybe some UVB light. I will show you the spectra in a moment. About from 205 nanometers to 320 nanometers. And with this light, we can perform, or nature performs, what is known as microscopic dissipative structuring. So we start with precursors, which may be rather simple precursors, which are supposed to have been on the ocean surface at the origin of life, like hydrogen cyanide, carbon dioxide, carbon monoxide, and water. And with this UVC light, we can produce these pigments. And these, I will hope to convince you, are the fundamental molecules of life. So, for example, we have the basis of DNA or RNA. These absorb very strongly in this UVC range and dissipate this light into heat. So the same thing that the pigments in the plants are doing today. So therefore, at the origin of life, well, we didn't have the life like we know it today. So we didn't have this dissipation of the visible and the near ultraviolet, except for some abiotic processes. But what we did have is these UVC pigments, which could dissipate the peak in the UVC light, which was coming into Earth's surface at the origin of life. So life's thermodynamic function then, at the beginning, was to convert UVC light into heat. And this is what I call the thermodynamic dissipation theory for the origin of life. And there are really three components of this. One is dissipative structuring. So the structuring of the pigments through dissipation, and I'll describe that in detail, what I mean in future slides. And dissipative proliferation of these pigments over the whole of Earth's surface, and evolution of these pigments into systems more complex, for example, arriving at the biosphere that we have today. So each one of these three components of this thermodynamic dissipation theory increases the photon dissipation. And that's what's the driving factor or the reason behind the origin and evolution of life. So before I get into dissipative structuring, it's important to describe a little bit the initial conditions at life's origin. In particular, how was the sunlight at life's origin? So this, we think, was about 3.85 thousand million years ago when life began. And at that time, the interior of the Earth was warmer because we had more radioactivity. It hadn't yet decayed, so we had a warmer interior. And that would imply that we would have had more volcanoes. And volcanoes then would have been spewing out similar gases to the volcanoes that are existing today, which are carbon dioxide, water, and hydrogen sulfide. We also know that the seas were warm, or the Earth's surface temperature was warm, about 85 degrees centigrade. And we know that from oxygen isotope ratios, which are found in some of the sediments which still exist, which are still in relatively good condition after 3.85 thousand million years ago. Now, the atmosphere then probably contained these gases which the volcanoes were spewing out, and probably a lot of nitrogen as it does today, because nitrogen is a very noble gas and it's found on all of the planets. There may have been also some methane and maybe some hydrogen through a physical process which is known as serpentinization, which is really the hydrolysis over these olivine rocks. But there would have been no oxygen and therefore no ozone, because oxygenic photosynthesis still had not arrived. So therefore the UV light must have been very intense. So what in fact did the solar spectrum look like at the origin of life? Well, we did some very simple calculations. These have also been done by others. I think the first person to do this calculation was Carl Sagan, and he published these results in the early 1970s. So if you look at the black curve here, that's the spectrum at the origin of life, this black curve. Now, to do this calculation, this is an estimate really. We took what is known as the Sun Through Time project, in which they look at proxy stars, G-type stars like our sun, of different ages. And they find that what happens is that although the spectrum doesn't change very much, as the surface temperature remains relatively constant, what does change over time is the size, the radius of the star. So the intensity does increase over time. And it's estimated that our sun has increased in intensity by about 30% since the origin of life. And so these, for example, the yellow is the spectrum at today of the sun, in which there is no UVC light. So the interesting thing, okay, and to do this calculation, I should mention, we take these atmospheric gases into account. We take their extinction coefficients, molar extinction coefficients. And we've also included the fact that we could probably form aldehydes through UVC interacting on these molecules. For example, formaldehyde. But we didn't have any oxygen or any ozone until about 2.8 thousand million years ago. We also take into account a little bit of scattering. We assume that the pressure of the atmosphere was maximum, and these graphs were taken with this maximum pressure of about 2 atmospheres. And that comes from looking at the raindrop marks in sediments available at that time. And we also include, after 2.8 thousand million years ago, some haze formation, which depends on these ratios of methane to nitrogen to carbon dioxide. But what I really want to point out is this little peak in the UVC. So this peak extended from about 205 nanometers all the way up to about 285 nanometers. And there was a little bit of light in the UVC range. And then again, about 310 nanometers, we have more light, incident light. So at the short wavelength region, the light was being absorbed by the carbon dioxide and hydrogen sulfide. And in the UVB range, it was being absorbed by the aldehydes, which were in the atmosphere. Now, the interesting thing is, if one plots the maximum of absorption, so the maximum in the spectrum, maximum wavelength of the maximum where the absorption is greatest, of these fundamental molecules of light, they all fit very nicely around this peak in the UVC. Not all of them, but most of them, many of them, maybe it's a better word. So here I have about 50 different fundamental molecules, those in the 3 domains of light, like nucleic acids, amino acids, fatty acids, sugars, vitamins, coenzymes, cofactors. And they all absorb very strongly, and they all absorb at these wavelengths. There's very little absorption in the UVB. And then again, in the UVA, we get further absorption of these fundamental molecules. So can we say that the fundamental molecules are really pigments in the UVC? And going even beyond that, could they be dissipative structures? And as I said, I will explain dissipative structuring in the future slides. But one thing I want to point out in this slide still is that the energy in the photons in the UVC range, this UVC range, are sufficient for remaking, that is transforming carbon covalent bonds, including double carbon covalent bonds. But there's not enough energy in these photons to cause ionization and therefore destruction of these molecules. So to explain dissipative structuring, there are 2 classes of structures in nature. One are these equilibrium structures, like crystalline structures or the solar system. And another type of structure, class of structure, are non-equilibrium structures. So the equilibrium structures arise through the minimization of the Gibbs potential. But non-equilibrium structures arise through the optimization of dissipation. And some familiar examples of this are the hurricanes, which arise through the dissipation of the temperature gradient over the system, the warm ocean surface, and the cold upper atmosphere. And Barnard cells, which are convection cells, which are also dissipating a temperature gradient over the system. And what are known as Belousov-Zhabotinsky reactor system, which is dissipating chemical potentials. So we have incident molecules coming in and product molecules going out. And this system is dissipating this chemical potential. Now, in the Barnard cells, we find a breaking of the symmetry, spatial symmetry, because before we have the Barnard cells occurring, we have simple conduction, and we don't find any change in the system, any observable change in the space symmetry of the system. But once we go beyond a certain Rayleigh number, which I'll describe in a moment, then all of a sudden we get this structuring, and this is called dissipative structuring. These structures are formed to foment the dissipation of the temperature gradient in the case of the Barnard cells. Now, these structures in the Belousov-Zhabotinsky reactor system, they're created to foment the dissipation of the chemical potential. And in fact, they change over time. So it's not only symmetry breaking in space, but also symmetry breaking in time. So these systems look like they're very much alive. Now, the conjecture today is that the fundamental molecules, for example, like DNA, were dissipative structures, non-equilibrium dissipative structures, but microscopic dissipative structures. And I'll explain the difference in the following slides. So first, to remind you about dissipative structuring, we'll take the typical example of the Barnard cell. In the case of the Barnard cells, we have a liquid which is sandwiched between two plates, a hot lower plate and a cold upper plate, and the liquid in between over gravity at a certain distance, L, the size of the system, and a temperature gradient over the system. Now, as long as we stay below a Rayleigh number, this is a number which depends on the parameters of the system, for example, the acceleration due to gravity, the viscosity, and the thermal expansion coefficients, and the temperature difference, the gradient of the temperature and the size of the system. As long as we stay below this Rayleigh number, then we get only conduction. But going over that Rayleigh number, all of a sudden we get this production of these cells, these Barnard cells, or these convection cells. And so in this case, we come into a nonlinear regime, actually, when we get to the Barnard cell. We go over the Rayleigh number. So if we increase, for example, the temperature gradient over the system beyond some value, we all of a sudden get this spontaneous in quotations, because really you need the temperature gradient of the system to create these cells. But in this nonlinear regime, in fact, we get more than one stationary state, and that makes sense because if you have a nonlinear equation, then you can have more than one solution to that equation. So for example, in the case of the Barnard cells, we could have the hot liquid coming up in the center, cooling off on the surface, and then going down around the edges. Or we could also have, which is another possibility, another stationary state, it's where the hot liquid comes up along the edges, cools off on the top, and then goes down in the center of the system. So in these nonlinear systems, and again, when I refer to nonlinear, I mean between the flows and the forces. So in the case of the Barnard cell here, the flow is the flow of heat, and the force is minus the gradient of 1 over the temperature, or just 1 over T1 minus 1 over T2, if you want to look at it in discrete form. So in this case, we have multiple stationary states, 2 stationary states in this case, and they are quasi-stable. They're not going to be globally stable because you have these 2 states, not only 1 state. And you can plot that on a diagram. So this, for example, on the right is the entropy production. And on the x-axis, we have some variable, which could be, for example, a Rayleigh number or the temperature gradient in this case. As soon as we go beyond some certain point in the Rayleigh number, then we get 2 possible solutions to the system. And it turns out that these 2 solutions have different entropy production. So for example, in the case of the Barnard cells, the solution in which the hot liquid is coming up in the center of the cell and then cooling off on the surface has greater entropy production than the solution in which the hot liquid is coming up around the edges. So we get these 2 different solutions. So the question then is, which one does nature prefer? Well, it turns out, especially for what are known as autocatalytic systems, and I'll explain that in a little bit of detail later. For these systems, the tendency is to increase the dissipation or increase the entropy production. But it depends on a stochastic fluctuation at this point, at the critical point. But generally, the tendency is to increase, but not always. It could also decrease because, as I say, for these nonlinear systems, there are many different solutions. We could also go down in entropy production, but the general tendency is to increase entropy production. Okay, so that was macroscopic dissipative structuring. Now I'll talk a little bit about microscopic dissipative structuring. And this is over molecular degrees of freedom. And in our case, they're going to be UVC induced. And these include, for example, isomerizations, tautomerizations, rotation around bonds, charge transfer, substitution, etc. Whatever molecular transformation which can be enacted through this UVC light. And the example I'm going to give is adenine. And this adenine, the production of adenine from the precursor, which is hydrogen cyanide and water, under UVC light was actually done in 1966 by Ferris and Oregon. So they doing this experiment actually in the laboratory, they found that they have important quantities of adenine being produced. Adenine, of course, is one of the bases of DNA, for those that are not familiar with the biology, and one of the fundamental molecules of life. So in 2009, we talked about this being dissipative structuring in an article that we published. And then in 2013, Boulanger et al., he did a time-dependent density functional theory calculation of the excited state potential energy surface of the system, going from hydrogen cyanide to adenine. And that was very useful for us. And some of the results in the density functional calculations are presented here in this graph about what exactly is happening. So we start with 4 molecules of hydrogen cyanide, and that forms to the minimization of the Gibbs potential, the lowest free energy structure, which is known as SysDAM. And SysDAM, in fact, absorbs a photon at 298 nanometers. And by absorbing that photon, it can do a rotation around this double covalent bond. So if it was just thermal chemistry or normal chemistry, that would never happen because, well, first, those carbon covalent bonds are very tightly bound. So it takes a lot of energy so you wouldn't get a statistical fluctuation sufficiently strong enough to cause the rotation around the double bond. And also it turns out that this transDAM, which is the result after the rotation, is of higher energy. So again, in equilibrium thermodynamics, the probability of occupying this state would be much smaller. It goes exponentially with the energy compared to the lower energy state, which is SysDAM. But as long as we have this flow of photons over the system, we can get a sufficient amount of transDAM in the system to allow it to evolve even further. And it does that by absorbing another photon, this time at 313 nanometers. And that causes a proton transfer. So one of the protons here, one of the hydrogens, goes to this nitrogen atom. Okay. And then this AIAC, I won't even try to pronounce what the chemical name is. They're very complicated chemical names. But anyway, AIAC can then absorb a photon at 275 nanometers to form this ring formation. And then through 2 more chemical reactions, or thermal reactions, we get to AICN, which is this system here, which absorbs very strongly at 250 nanometers. And then with another hydrogen cyanide, so 5 hydrogen cyanides in total, then we can form adenine. Now, it turns out that adenine has a very large molar extinction coefficient, 15,000 in these 1 moles, and molar, inverse centimeter units, which is not as strong as chlorophyll, but it's getting up there. It's very close. And not only does it have a very strong molar absorption coefficient, it also dissipates its energy to the ground state very quickly through what is known as a conical intersection. So in the schematic diagram below, I have a conical intersection. And the 2 axes are really can be considered, for example, the coordinates of the system. Of course, there are 3n-1 coordinates of any system, where n is the number of atoms in the system, in the molecule. But here, this is just a schematic diagram. So we have, for example, 2 coordinates of the atoms in the system. And the blue is the electronic ground state. And when the system absorbs a photon, it goes to this upper state, excited state. And it turns out that when it does that, it actually deforms. So one of the hydrogen, this is actually one of the carbon and the hydrogen, come out of the plane. So the bases are normally planar, but when they are excited, they become non-planar. And because of that deformation, it turns out that the vibrational states superimposed on the electronic states become degenerate between the ground state and the first excited state. And that is this formation of this conical intersection. So the energy can go very quickly from the excited electronic state to the ground state. In fact, it does that in about 10 to the minus 12 seconds, so in even less than a picosecond. It's a picosecond, but less than a picosecond. Now, the interesting thing also is that, as I mentioned, we can go up in energy because we use the energy in the photon for these photochemical reactions. And that's the same as we could say going downhill in entropy. So we're organizing, the system is being organized. And why is it organized? Well, it does that just the way a hurricane does. It organizes to increase the dissipation, which I'll show you in the following slides. So here we have on the right, at the bottom, we have a hurricane, this is Irma in 2017. It's formed near the equator. And we see that the hurricane is growing in size, and it's growing because it's moving over warm water. So it's the water, the temperature of the water, surface water, which is causing the hurricane to travel in a certain direction. So that's not the only factor involved. There's also winds involved, but one of the major factors is the temperature of the ocean surface. So we see it growing from a tropical storm to a hurricane of 4 or 5. I'm not sure what this one was. So we see that the system is over time increasing in size, increasing in dissipation, and it's being steered by the gradient, the temperature gradient, to some extent at least, also by the winds, as I mentioned, but it's being steered by the temperature gradient. Now, if we look at what's happening here in the production of adenine, it's really the same thing which is happening, but on a more microscopic scale. So as I mentioned, system, this molecule here, which I call molecule 2, it's absorbing at 298. So that's about here on the spectrum. Remember, this is the UVC light arriving at Earth's surface at the origin of light. So it's absorbing at 298. And then it goes to 313. I lost my pointer, but okay, here it is. So at 313, it's absorbing this light. It's absorbing a little more because there's more light available. And then it goes in the case here to 275 nanometers here. That's this AIAC. So this molecule here. And that's absorbing a little more light, and it's also its molar extinction coefficient is increasing, at least from this one. And then it goes to AICN, which is absorbing at 250 nanometers. And then finally it goes to adenine, which is absorbing at the peak of the incident UVC spectrum at about 260 nanometers, just a little below that. And it has a very large molar extinction coefficient. So this is a very strong dissipative structure. So this is like a hurricane 4 or 5. I'm not sure if there exist hurricanes of 5, but so it starts here as a tropical storm, and then it goes to hurricane 1, 2, 3, and 4, where it's absorbing and dissipating a lot of incident light. So this is microscopic dissipative structure. So the fundamental molecules then are dissipative structures. They take a photon in the UVC and dissipate this into many infrared photons. And they do this in about 10 to the minus 12 seconds, picosecond or under. And that's very similar to what a hurricane is doing, because it's taking infrared light from the ocean surface, at the temperature of the ocean surface, about 28 degrees centigrade, and it's dissipating it at the cloud tops at about minus 15 degrees centigrade, and emitting it this energy into space in many more infrared photons. So it's also a dissipative structure. This is macroscopic and this is microscopic. Okay, so I talked a little about dissipative structuring, gave you the example of adenine. We have also done it for guanine, and this has been published also. And this was done in collaboration with my student, Claudette, who's now at the University of Calgary. And so now the second element of the thermodynamic dissipation theory for the origin of life is the proliferation of these pigments over the ocean surface. So let's assume that we can do this for all of the molecules. We think we can. We've done it for adenine and guanine, and also for fatty acids. And we're going to do it for the pyrimidines also, we hope. So let's assume that we can build all of these structures through dissipative structuring of this UVC light, and that we have these elements available on the ocean surface, and that we can form single strands through the piling up and in interaction through the pi interaction, and form DNA, single strand DNA. There's much more I have to talk about, of course. For example, the ribose and how the phosphates become attached and all that, but most of that can also be done by UVC light, which is very interesting for us. But we haven't looked at that in detail yet. So I'll take the example of DNA, and this proliferation or replication has to be enzymeless, because of course we didn't have these complex enzymes which are needed today to reproduce DNA. For example, today we need about 20 enzymes, very complex enzymes, to reproduce DNA. So in place of enzymes, we came up with the ultraviolet and temperature assisted replication, which we call UV-TAR for short. So as you may recall, the surface temperatures were very warm, about 85 degrees centigrade, and they were cooling throughout the Archean. So at the origin of life, they were 85 degrees centigrade and then cooling throughout the Archean. So if we had single strands of DNA floating on the ocean surface, we can imagine that as the temperature cooled sufficiently enough, for example, and this is very interesting, that for these short strands segments, about well, less than 200 base pairs, the denaturing temperature is about 85 degrees centigrade, and that turns out to be what the Earth's surface temperature was at the origin of life. But as we go below that denaturing temperature, then single strands would come together, or pieces of single strands would come together and form these double strands. And then as the ocean surface temperature continued cooling, they wouldn't be able to denature again, so we wouldn't have replication. But we have the sun coming up in the morning, warming the ocean surface through infrared light. We also have this UVC light coming in from the sun. And this UVC light can absorb on single-strand DNA, on double-strand DNA, sorry, and cause it to denature. So this is not hypothetical. In fact, we measured it in the laboratory and we published an article on that. And the interesting thing is when this UVC light comes in, so if we're close to the denaturing temperature, but below the denaturing temperature, a flux of UVC light can cause the system to denature. And when it does denature, the interesting thing is that it opens up like an umbrella. So the bases are more exposed to light and they absorb more. So this is called a hypochromic effect, and the increase is about 25 to 30%. So after in the afternoon, when the ocean surface temperatures may be 4 or 5 degrees warmer, as it is today, you can measure it today, and it turns out to be about 4 or 5 degrees warmer, and we assume we had a similar warming of the ocean surface at the origin of life. And with these UVC photons, we can cause denaturing. And then at night, when the ocean surface temperature cooled, we could have renaturing. So the formation of double strands, where we once had only one single strand, now we get 2 double strands. And this has been performed in the laboratory, not by us, but by others using magnesium ions to control the reformation of these double strands. So the interesting thing here also is that this is template directed, so it's an autocatalytic photochemical reaction. And that replication is tied to dissipation. So the better that these systems are of absorbing ultraviolet light and dissipating that light into heat, the greater probability that they will denature and therefore replicate, because denaturing is the first step in denaturing. It has to first denature before it can replicate. So DNA replication is related to photon dissipation or entropy production. So remember, in our scheme, everything has to be related. We have to give a reason for the occurrence of everything, and it has to be related to increases in entropy production. So this is the experiment that we did in my office, actually, just on the desk. We had a deuterium light, and we had a temperature-controlled cuvette holder. And in the cuvette, we had water or PBS buffer and a DNA sample. And we had a spectrometer to measure the absorption and another spectrometer to measure the scattering, which is very small for DNA. In fact, we neglected it in the analysis of our results. And we have a water cooling, and this cooling through this temperature control system allows us to increase or decrease the temperature very rapidly, and also make sure that the temperature remains very stable. And everything is connected to a laptop. So these are actually our experimental results. This is the absorption spectrum for 25 base pair synthetic DNA. And we produced this DNA in our institute or in another institute of physiology in the same university. And we designed them so that there was no two thymines next to each other, because we know we can get production of dimers, thymine dimers. And so here what we're doing is just with measuring the spectrum, the absorption spectrum. So it's a function of wavelength. And as I mentioned, at 260 is about the maximum, 258, actually 259 for DNA, this is 25 base pair DNA. We calculated or determined the spectrum at different temperatures from 30 degrees up to 80 degrees centigrade. So this system actually denatures about 75 degrees centigrade. So when we're at 30 degrees, we have double strand, and when we're at 80 degrees centigrade, we have single strand DNA. And we get this, of course, what we're seeing here, this increase in the absorption is due to this hypochromic effect. And now what we did actually was around each temperature, we took a 3-degree bin. So we did the lower and the upper of that bin. And that's why we see 2 lines for every color, because we have a 3-degree bin here. And then we took the difference spectrum from the hotter temperature, lower temperature, 3 degrees difference, and plotted that as a function of temperature, or for the colors as a function of temperature and a function of wavelength. And this is these plots in the bottom of the graph. So this is the difference spectrum. This corresponds to these for a 3-degree bin around these temperatures. Okay, so this is thermal denaturing, and that's nothing new. In fact, they use it in the PCR in the laboratory, so it's very well known. Now, what we wanted to see is if we could produce UVC light-induced denaturing. So what we did here, for example, on the left, we took salmon sperm DNA in water, in pure water. We took the system up to a temperature about 40 degrees centigrade, and then maintained it at that temperature. And then after an hour, when the system stabilized, so we had the light on during this hour, and when the system stabilized, we turned off the UVC light. And then we waited half an hour, and then we turned on the UVC light. So whenever the up arrows are, we're turning on the light, and the down arrows correspond to us turning off the light. But this is at a fixed temperature, remember, so this is 40 degrees centigrade maintained throughout the experiment. And you can see that every time we turn the light off, the absorption goes down. When we start again, turn it on, it goes up and down. So we're seeing this system which is naturing and denaturing. So this was for salmon sperm DNA, and we find the same thing for the synthetic DNA. And this is the results for 25 base pair synthetic DNA. Now, this is the solid lines here are for double strand, okay? and the dotted lines are for single strand. So we took also single strand DNA to see if, just as a control, to see if what we're really seeing is due to the fact that we're denaturing and not some other experimental effect. So that the single strand was the control. So when we have the double strand DNA in this PBS buffer, and we fixed the temperature, these are the different temperatures here on the right, upper right. And we took the different spectrum, which is from the light on, okay, the light at the last point, the light on spectrum, to the beginning of the light on spectrum. So the different spectrum here. So this is just as a function of wavelength, what this looks like. This is integrated. I should have said this was integrated over 255 to 265 nanometers. So we're integrating around the peak here. This is the integrated absorption around, integrated over this wavelength range. And so we find the same thing, and we find these now. The interesting thing is for single strand DNA, we found a dip. So the different spectrum, so it decreases, the absorption decreases after half an hour of light on. And the answer that we think is the reason that it's doing that is that it is forming dimers, but they're not thymine dimers. They may be dimers with thymine or cytosine, because we did have some of those in the system. So, and that actually reduces the absorption. And if you look at the data which is available in the literature, it is around 270 nanometers where we get a decrease in absorption if we form these dimers. So we think in the single strands with the UVC light on, we are forming these dimers. And that's why we get a decrease. Okay, but this is the different spectrum, and this is only for UVC light. We're not changing the temperature. Each of these is at a different temperature, but for the whole experiment, the temperature is kept constant at that particular temperature. So now integrating those curves, we can determine what is temperature. At a given temperature, we can determine what is the overall effect of UVC light-induced denaturing. And we plotted that on this graph, which is the effective denaturing in percent per hour on the y-axis, and the temperature on the x-axis. So as I mentioned, for this system is 25 base pair synthetic DNA. The denaturing temperature was about 75 degrees centigrade. So as we go towards the denaturing temperature, we get an increase in light-induced denaturing. So as we get closer and closer to the denaturing temperature, that's when the UVC light is most probable to cause denaturing, and that sounds relatively obvious because the system is weakly bound at those high temperatures, and so any effect could cause it to denature. So what is happening then is we have a photon coming in, which is being absorbed on one or two of the bases, and that energy is being spread. Actually, we assumed that, well, we thought that this was probably due to heat being created locally, but it turns out that in water, that heat is dissipated so fast that in fact, what we're probably seeing is proton transfer from one of the bases in the pair to another base. And that reduces the stacking interaction between two consecutive bases, and that reduces the strength. So as long as the photon is absorbed near the ends, the frayed ends, okay, these ends become frayed, then we get UVC induced denaturing. And so once we get a few of these separated, then the whole system just unzips very quickly. Okay, so this then was to explain this part, which is UVC induced denaturing, and how we can get replication through dissipation of photons. So now I want to get just a little bit to talk about the homochirality, because as we know, life is homochiral. So DNA is right-handed and the amino acids are left-handed. So how did this come about? This is one of the important questions in the origin of life research. And we really don't have a good explanation so far. I know that Noemi is working on her explanation, which sounds very intriguing, using spin polarized particles coming from cosmic rays. But here I want to give you a proposition, which is using this ultraviolet light at the ocean surface. And the fact that we have a dichroism spectrum, so that means we're absorbing a little bit more, for example, for right-handed DNA, we absorb a little bit more right-handed circularly polarized light than left-handed circularly polarized light. And that's given by this, what's called a dichroism spectrum. So here, when it goes negative, that's when we're absorbing more right-handed circular polarized light. Now, these are for different sizes DNA. And now the interesting thing is if we connect a tryptophan, so tryptophan is an amino acid, which is an aromatic amino acid, and it also absorbs in the UVC range about 280 nanometers. Now, when we include tryptophan in the system, then we get a much, much greater circular dichroism. So yeah, I put this on the same scale. I didn't want to put this on a logarithmic scale, but here I just note that you have to multiply these values. So read off the values here and multiply them by 4 to get the correct value. So with tryptophan, which could be acting, for example, like an antenna molecule, we get much more circular dichroism. Now we have to do the conjugation over the incident spectrum, incident UVC spectrum, and over the absorption spectrum of DNA, and that gives you these graphs. So they're similar, but they're just integrated over the UVC and over this wavelength range. Over the wavelength range for the incident and the absorption spectra. Okay, so how do we get circularly polarized light on the ocean surface? Well, it turns out this has been known for some time, maybe 50 years now, 60 years. And in fact, it turns out that the region of greatest polarization in light in the biosphere is actually just at the surface underneath the surface of the water bodies. So what happens is the light from the sun comes in, it's scattered off particles or molecules in the water, and that causes linearly polarized light. And then it becomes totally internally reflected, and that gives rise to circularly polarized light. And it depends on the direction of the view, but it's either right or left-handed. And depends, for example, if the sun is in the morning or in the afternoon, you get a different polarization from a particular position. So this is how we think that what would have been happening now. So in the morning, let's say we get more left-handed due to this effect of polarization, we get more left-handed circular polarized light in the morning. But in the afternoon, we get more right-handed circular polarized light. Only a little more, maybe 5 or 7% more. And so we assume that we have a racemic distribution of the DNA, both right and left-handed. But now it's absorbing these photons, polarized, circularly polarized photons, and denaturing. But so what is the difference? Where does the symmetry breaking come in? Because in the morning it's left-handed, a little bit more left-handed, and in the afternoon it's a little bit more right-handed. Well, the symmetry comes in because of this graph, which is the denaturing rate as a function of temperature. And the fact that in the afternoon, the surface temperature is a little bit warmer of the ocean, maybe 4 or 5 degrees warmer. So we get a little bit more denaturing in the afternoon with right-handed circular polarized light as we would with left-handed circularly polarized light in the morning. And so you can plug all those numbers in for the amounts of circular dichroism. And we can do this calculation where we're assuming that the number of left-handed DNA is equal to, at a given generation, I, or a given day, let's say, is equal to the number of left-handed in the previous generation, multiplied by some factor, which is just a parameter C, which corresponds to the quantum efficiency for denaturing, which we can assume some value for that. And then these probabilities of absorbing a photon, for example, PLR is the probability of absorbing a right-handed photon on left-handed DNA, and so on. So we can create this recursion relation for the number of DNA which are left-handed or right-handed, those which get reproduced because we need the denaturing for reproduction. It's the first step in reproduction. So plugging in these values and assuming, for example, that we need about 1,000 photons before we get a short strand DNA to denature, and the fact that we have about 5% more right-handed circularly polarized excess light in the afternoon at the ocean surface, and then we have only 0.07% right-handed circular dichroism for the right-handed DNA. No, it corresponds to this value, which is given in the literature of 4/6,000. And we make another assumption that we get no denaturing in the morning. That's just to simplify it, but we could have done it. There's no reason why not, and probably we'll do that in the future. But to simplify it, we assume that we get no denaturing in the morning and only denaturing in the afternoon. And we assume that we have initially a racemic distribution of the both left and right-handed, so equal. And we set the absorption of left-handed circularly polarized photons on left-handed DNA equal to 1. And then you can convince yourself, I won't go into it, but you can convince yourself that these are the correct probabilities for the other cases. For example, right-handed circular polarized light absorbing on left-handed DNA. And then we can calculate the chirality for the system using these recursion relations. And that's just the number in a particular generation of left-handed minus the number in the right-handed over the sum of the two. And then plot that on this graph as a function of time or Archean days. So very interestingly, we find that we get very quick, if we include tryptophan in the system, because that gives us a much greater bright circular dichroism of the system. In only about 140 years, we get 100% chirality or homochirality. We get homochirality only about 140 days during the system. And these other graphs are for without the tryptophan and for the different size DNA. And the graphs which are jagged here are because we included an energy threshold into the system so that we don't get any denaturing unless the energy is above some point. So it's not just continuous probability, but only when we go beyond that expression value of the energy which is needed for denaturing of the system. And we can see we get much quicker homochirality occurring when we include that energy threshold in the system. Okay, so I've talked about dissipative structuring, dissipative proliferation. Now I want to talk a little bit about dissipative evolution. So there are three elements in this, and they correspond to this, well, what we've already mentioned before, that we need a generalized thermodynamic potential. I'm just going to turn on the lights here because we're getting into nighttime. Okay. And so this,
I don't know if you have many slides left.
you know, I'm finishing. This is the second last one.
Okay, thanks.
Okay. So there are three elements which are required. One is this generalized thermodynamic potential. So this is the force over the system. And of course, this comes from the environment, and this is the sunlight over the system and the coldness of outer space that defines the temperature gradient, if you like, or the photon potential. And that gives rise to a generalized thermodynamic flow, and that's the flow of UVC photons over the system into producing these infrared photons. Okay, so we have also spontaneous dissipative structuring, as I mentioned, producing adenine, and that's absorbing and dissipating the light. Now, the third component that we need that I talked about a little bit earlier is the nonlinearity in the system. And that gives rise to multiple stationary states. So that's what allows the system to actually to evolve. And I gave the example of the Bernard case, where we have two states, the liquid coming up through the center and cooling off on the top and going down around the edges, or the hot liquid coming up around the edges and cooling off and going down in the center. So there are two stationary states there. And as I mentioned, the state that nature tends to take is the state with greater entropy production. So this is just another schematic diagram. So here we have the entropy production on the y-axis. And we have different variables for the system, which depend on the hierarchy. So for molecules, for example, it could be the concentrations of the different molecules, these intermediate molecules in the system. And for ecosystems, it could be populations of species. And in the biosphere, it could be populations of species types. So here we have a case in which we have three stationary states, so three solutions to the equations, and how the system evolves from one to another. Well, when it gets trapped in this local maximum here, we need a fluctuation or perturbation, either externally or internally, to cause the system to move to another state. So, and it does this when the system is close to these, the boundaries of these peaks, which are these critical points. If you remember that to get to a stationary state of the Bernard cell, we need to go through a critical point. So these are the critical points in the system. This is in two dimensions instead of one. And so the system can go through stochastic fluctuation, can go from, for example, let's say we start on peak 1, it can go to peak 2, and then to peak 3. And the tendency would be to go, if the fluctuations are really stochastic, would go towards the greater peak, because it has a greater basin of attraction in general. And that's particularly the case when we have autocatalytic systems. So for example, autocatalytic chemical systems, you could imagine that if you don't have the temperature, let's say that for that system, we have one of these axes is the temperature. If we don't have the temperature quite right, as long as it's autocatalytic, then the reaction will go forward. So that increases the basin of attraction of these systems. And that's why the general evolution of the system is toward greater entropy production for these autocatalytic systems. So, okay, so that's evolution of the system. So in conclusion then, life's function is to dissipate sunlight into heat. And it does that through the dissipative structuring at the origin of life of these fundamental molecules. And I gave the example of adenine, but we've also done it now for guanine and for fatty acids. And I gave you a little bit about the dissipative proliferation over the whole of Earth's surface, which is, of course, going to increase the dissipation, the global dissipation. And then I mentioned dissipative evolution, how the system, through a statistical or stochastic fluctuation, the system can move towards stationary states of greater entropy production. So therefore, I hope I've given you a reason to believe in this non-equilibrium thermodynamic explanation for the origin and evolution of life. And before finishing, I'd just like to thank my collaborators and students who have been working with me for maybe over 10 years now on this project. Alex Simonov from Cyril and Methodius University in North Macedonia, and Oliver Manuel from the University of Missouri, Jose Manuel Nieto from the University of Havana in Cuba. And my students here, we have now like 16 students who have come through this, done their thesis on this. Some of these students are here today, Andres and Julian here. I'm not sure if the other students are here. I can't see their names, but they've all been doing particular work on these topics. So I'm very grateful for their help and also for the financial support of the GAPA UNAM project. And finally, I have a book which you can get on my research page, which describes everything I've talked about today in a little bit more detail. This book was published in 2016, so we have more results. And those results are published in a number of papers. We have actually now published 22 papers on this theory. And those can be all downloaded also from my research page. And they're free of charge because I like to go for open access publishing. So thank you very much, and I appreciate very much your questions or comments, criticisms. Thank you.
Karo, I have a question.
Sure.
Can you hear me okay?
Yes, if you come a little closer, maybe I hear you even better.
Okay. Is this better?
Yes.
Okay, good. So I'm a physicist, and I happen to be working on molecules of water. So some of the biology is not well known to me, but it definitely seems that your ideas associated to the understanding of fundamental molecules emerging from dissipative processes is very reminiscent of what someone was doing condensed matter for understanding spontaneous symmetry breaking in the production of dissipative topologies such as vortices and superfluid.
Yeah.
And so I'm involved in some of that, but I was fascinated by your talk because I never extended those physics ideas to fundamental molecules as you have. So that's beautiful and astounding. So now, secondarily, you also mentioned that this process of denaturing leads to replication, which of course has the byproduct of increasing entropy. And that also, we of course see this replication in DNA. So I guess one of my fundamental questions is, can we extend this replication concept from denaturing to actual replication of more fundamental aspects such as molecules, say water?
Yes, in fact, with the student Julian who was here with me, we did the idea of a complex system of tryptophan together with DNA. Okay,
Yes.
so tryptophan also absorbs very strongly, about 280 nanometers. And the trouble with tryptophan is that it doesn't have a conical intersection. So it doesn't dissipate the energy very quickly. It actually fluoresces, which has a much greater lifetime, like many orders of magnitude, much greater lifetime. So however, if tryptophan could couple with DNA, then it could pass its excited state energy onto DNA through energy transfer, as long as it's close enough, and then use the conical intersection of DNA, of the bases of DNA, to dissipate that energy into heat. And so the system as a complex is much greater dissipator than system separated. So that's one example of complexation. And in fact, the interesting thing about that is that there was in fact a stereochemical era for DNA or RNA and the amino acids, because a lot of these amino acids have chemical affinity to DNA. So just bare DNA and amino acids, they will have chemical affinity to DNA, but not only that, but to their codons, to their particular codons. So you could imagine if we have this system, this UV-TAR system operating, then those DNA which coded for tryptophan would have these antenna molecules sitting on top of them. So they would absorb more photons, so they would denature more quickly or more often. And so we would get more replication. So that that's a further step, no? But
yes.
We've also considered it, for example, we can say today in the biosystems that we have this complexation of biotic dissipative systems with abiotic dissipative systems, like the water cycle.
Yeah.
And also the animals with the plants and the cyanobacteria. So we can find these couplings in many different areas, and these couplings always give rise to greater photon dissipation. So yeah, so we have thought about that, and in fact, I've just published this year article about natural selection and going from molecules to the biosphere. So
very interesting.
natural selection in terms of thermodynamics, non-equilibrium thermodynamics.
Yeah.
Yeah, so I'll send you that paper if you send me
Yeah.
an email.
Yeah, I'll send you my email. And if you could write your email and the paper that you just mentioned on the chat, that would be very useful. So I'm Carla Galdames, and I'll put my email here.
Sure, I didn't quite hear that, the final.
Yeah, maybe
I'm putting my email here.
Okay,
we should ask people in the room if they have questions. So maybe. Yes, Dave?
Can you hear
yeah,
me?
that's very interesting also about the formation of water molecules. You're talking about water structures, no?
Yeah, that is what we're
Microscopic
doing.
water structures.
Yeah, I'm looking at both.
Dissipation.
Yeah, looking at possible proliferation of water molecules, and then also looking into ground state of water and of itself by understanding enhancements of 0-point energy. So
See, and this is, you're dissipating infrared light, I imagine, there then, no?
right now, I'm primarily working on models, on computer model simulations.
Oh, okay.
I hope to expand it to water and light. So I should be in touch with you. If you could write your email, and I'll write you an email.
Okay, great. Yes, thank you.
Thank you. Have a wonderful afternoon.
Thank you.
Talk to you later, Car.
Yeah, talk to you later.
Okay, can you hear us, Karo?
Yes.
So we have a question in the room from David.
Yes, I was interested in your hyperchromic effect that you're using to demonstrate a denaturation. Now, you pointed out the denaturation of double-stranded molecules, DNA in particular, produces 20 to 30% increase in the UV absorption around 260. And I do that sort of measurement myself, so I agree with that. But in your light-induced, UV-induced, what you are interpreting as hyperchromism doing to kind of a melting, those were, as I remember the slide, relatively small changes in the UV absorbance compared to what you would expect from a complete melting of the chain. Is that correct?
Well, you mean these curves?
So the
Yes.
transfer, I see 4 cycles of light and then dark, and I see a hyperchromicity of about 0.68 to 0.67, yeah, in each of those. So how complete a melting does that represent? My guess is that that is not a complete melting of the duplex molecule.
Well, we took that into account when we did these calculations. So I have to go back to the data to be able to explain it completely to you. But this is like the effective denaturing, which is like taking this hyperchromic effect. We don't know, for example, this might be a little bit of denaturing on all of the synthetic DNA, on all of the individual. No, it might be a little, or it might be some of them completely, and others nothing. So we don't know exactly what is. But the point is that once you get near to the denaturing temperature, it only takes a little to cause complete denaturing. So, and especially if that happens at the frayed ends, at the ends of the DNA, if they happen here or here, then we get a fraying. And if we're very close to the denaturing temperature, then only the stochastic variations due to the temperature in the system will cause the whole system to denature.
Right, I agree with that. It should be more prominent at the higher temperature, near melting. And of course, you point out that there's a fraying effect that starts the process.
Yes.
Right down at the bottom. So I agree with that too. Now, let's just consider for a moment how the absorption of light energy could affect the denaturing. it occurs to me that there's two kinds of forces or energies holding DNA into a duplex structure. The one we all think about is the hydrogen bonding, but there's
Yeah.
also stacking energy.
That's not the important one. It's the
There is a
stacking which is
substantial
important.
amount of the stabilizing energy.
Right.
So, somehow the absorption of the UV photons is changing one or both of those to promote the melting that you're seeing. So
Yeah.
I wonder if you have thought about what could be happening that would affect either the hydrogen bonding or the stacking.
Yes. So in fact, we did some simulations of this. We thought at first we thought it was probably local heating, but it turns out that because of the water and the vibrational interaction between water molecules, it goes very fast. So it probably is not that what is happening. So let me see if I can get to. I have some more data here. I'm not sure if it's here. So, oh, I guess I don't have it here. I don't have it here. But what actually happens? What we think now is the reason for the denaturing is that in this hydrogen bonding, we can get a proton being transferred from one base to its pair to the other base. And when it does that, so that's just like it's another minimum in the free energy. So it's relatively, it's metastable. But when it does that, it destroys the stacking interaction. So that makes the system weaker. So we think it's actually photon-induced proton transfer of these protons, which are involved in the hydrogen bonding between the two bases, which is causing the denaturing. We would like somebody to do a theoretical calculation there, see if that could be a possible reason.
Thank you.
But as I said, first we thought it was just local heating due to the absorption of the photon and the production of infrared energy, light, but that doesn't seem to be the correct answer. It's probably proton transfer. So when you get the hydrogen protons transferred from one base to its conjugate base, then that reduces the stacking energy of the system. And that's probably what leads to denaturing. But so far, it's just a guess that has to be analyzed in detail.
So Karo, thank you so much. I will just, if people want to stay and chat more, I mean, I think.
Thank you very much.
We can also enjoy some food if you want, that is at the back, and you can, ask more questions.
Yeah, does that work for you, Karo? If we have a little bit more informal setting at the time?
Sure. Yeah,
Okay.
you can have a wine and dine session right now, and I'll take some hibiscus water. And anybody who has questions informally, great, or they can also email me.
Yes, so let's thank Karo again for this wonderful talk. And yeah, so let's have a little bit food, and if you have more questions, feel free to ask.
Also on the
Okay.
chat, I guess. I don't
In-
know.
I don't think there are
There are-
questions on, no, there are no questions on chat right now at the moment. Yes.
Karo,
Yes.
it's me again.
I can
I
see-
have a follow-up question.
I'm going
Okay.
You probably know that Nomi and I and Dave Krieger, who was here,
had to leave.
had to leave, right? We're doing experiments, so rather close to the sorts of things that you were doing. We are using synthetic DNA. We're following UV damage to the DNA.
Yeah.
And for differential effects of circular polarized light on the helical structure.
Okay.
So that's why we've been getting into some of the same kinds of measurements that you are doing. What we see, besides this sort of reversible denaturing that you reported is a permanent photo bleaching of the bases. Now we're using laser light at 266 nanometers, the UV range, and we can watch over time of about up to 40 minutes of these laser flashes that are doing the photochemistry. And we can see the UV absorbance decrease from the original rather rapidly, all the way down to maybe only 10 or 20% of the UV absorbance left, as though all of the bases are in fact going through some photochemical process that I would call bleaching, for any better term. So I wonder if you have seen any bleaching that goes on in your experiments.
Yes, I think I mentioned a little bit about that here when I was talking about the difference between single-strand DNA exposed to UV light and double-strand DNA. Because when we expose single-strand DNA to light, of course, we're not going to get any denaturing. It's already denatured. And so we don't see any increase in the absorption. But what we do see is a decrease. And that corresponds to about 275, 272 nanometers. And that is what we think is the formation of thymine dimers, or in this case, dimers between other pyrimidines. So, because in our synthetic DNA, we didn't have any thymine dimers. We designed it that way.
Right.
But maybe in your case, if you have thymine dimers, then you're going to see a decrease in the absorption, a negative effect here, shining UVC light on it. And of course, that also depends on the wavelength of the light and also the flux, the intensity of the light. Now, so that's what I think you call bleaching.
Yeah.
Is when we have this dip due to the formation of these dimers, thymine dimers.
Yeah, thymine is certainly the most labile.
Yes, it's the most probable.
If they're neighboring in a chain, then you do get thymine dimers.
Right, if they're consecutive.
Yeah.
But the same thing can happen not only if you don't have any thymine dimers, like in our case, but if you have thymine and cytosine, for example, next to each other, they can also form a dimer. The probability is much smaller, I think like a 1% probability compared to the two thymine together. But they can also form dimers, and that decreases the absorption of DNA.
Okay.
But
good.
now the interesting thing is, I also want to mention that occurs at about 270. So you get maximum bleaching at about 270 something nanometers. Now, if you have light over the whole spectrum at about 220 nanometers, you get a reduction in the dimers. So the dimers, they come apart again. They become separated, the bases.
Yeah, I've read about that. It's quite confusing.
Yeah, well, that happens at a different wavelength, 220 nanometers.
So
So
you have to heal the dimer by
yeah,
another wavelength.
it reverses the dimer back to its original state of two base pairs. So if you have a spectrum and not only one wavelength, then you can get this de-bleaching, if you like. And so the system will come to a stationary state in which you have a certain percentage of dimers and then stay fixed. So you could try that also with a spectrum, no? With the light, for example, from deuterium, like what we used here. We have a spectrum, and let's see if you get any reduction in the bleaching.
I just had an idea that might be fun to follow up. UV 260 nanometers produces the thymine dimers, and that gives a human body a sunburn. You know, that's a killing dose of UV at the UVC. But I wonder if it could be healed if we rapidly give a 275 nanometer to break up the dimer.
Okay, 275 actually creates the dimer. 220 breaks it up.
Oh, okay, good. Well, I'll give you that. So maybe we use a 220 light to heal the sunburn, as it were.
Yeah. And of course, it doesn't have to be from a laser because with a laser, you get only a single wavelength.
That's right. That's right.
But the absorption is very broad, as you can see here on DNA. You know, we're absorbing over 20 or 30 nanometers. And so it's very broad absorption. And that also indicates that it dissipates very quickly through the Heisenberg uncertainty principle. So if you get a large width in delta E, then delta T has to be very small. So yeah, it would be interesting to see what your results are at 220 nanometers or with a light which has the whole spectrum. So that's in fact one reason why we believe that this light was not very dangerous to the origin of life, to life at the beginning. Many people, even Carl Sagan, look for ways to shield life from this UVC light, no?
Yeah.
But it seems like it's not necessary, and that life needed this UVC light because with thermal chemistry, you cannot get these molecules produced even at very high temperatures, you know, like they found in the hydrothermal vents.
Yeah, I try to reassure people that worry about that, you know, and the easiest answer is just to say, yeah, there is shade there if we need it. But your answer is a better one, that it just isn't that damaging.
Yeah.
But isn't that damaging for this particular molecules?
Say again.
I'm saying it's not damaging for adenine, guanine, for the particular bases we are using, but maybe this is how life selected these ones, because other ones will radiate and not dissipate through this conical intersection.
Could be. It's a nice thought. You want to express that to Karo?
Carol, did you hear me?
No, I can hear you. If you come a little bit forward, I can hear you a lot better. Sorry.
I was just saying that I mean, this particular basis that we have, they excite through this conical intersection.
Yeah.
But other like ring-like molecules that were also present, maybe they were not exciting through this. So maybe this is the way life selected actually these 4 bases.
Yeah.
Because they are just this exciting without radiating a photon, so you don't make damage to the molecule. This is what you explain. While other rings would fluoresce, right? So people are always asking why life chose this particular molecule, why there was this selection of only this basis and not other types of molecules that were also present. So do we know if there are like other, like when you produce the from the very simple molecules you had at the beginning, this HCN, CO2, and etc. What are the products that you're producing? I mean, are there other like bases that you could produce but will not actually disexcite through the conical intersection?
Yes. Well, as you see on this graph, we plotted, there's like 50 fundamental molecules, those in the 3 domains. And they all absorb very strongly. Not all of them, like tryptophan, for example, doesn't have a conical intersection. So that's the interesting thing, that these molecules all arrived around this region. But if tryptophan has some kind of and it does have a chemical affinity to its codon on the DNA, then it can transfer the excited state energy to the DNA and use the conical intersection of the DNA. So even though not all of these fundamental molecules have a conical intersection, we believe that there's the complex of them with DNA allow the system to be a greater dissipator and therefore more selected or more replicated because, well, for this example I gave, they denature easier, more often. So yeah, not all of them have a conical intersection. We're just starting this, so we don't well, we've been doing this for more than 15 years now, but we're just starting to look at each one of these molecules. We have done adenine and guanine and fatty acids. So those do have a conical intersection. And the purines also do have, but we haven't produced the this scheme for production of, but there are experiments that produce the purine, the cytosine or the thymine or cytosine.
So do you know someone that did that for thymine? I would be interested to see.
That did what? Sorry?
The same plot that you have here for adenine, but do you have it for thymine, or do you plan to do it for thymine as well?
Well, you see, what we can do is now that, because many of these molecules, for example, guanine and adenine, they start with the same process. So basically the same process, and then they diverge at some point.
I see. Okay.
And use other molecules. So basically we have the beginning of them, but not the ending of them. So we have to, that's one of our projects, is to look in the literature for work like, for example, what Boulanger et al. Have done for adenine, which is calculate the excited state potential energy surface. And so that will allow us also to, if they've calculated the molar absorption or extinction coefficients, that will allow it to us to do other fundamental molecules. But we haven't done that for other than adenine, guanine, and fatty acids.
Okay.
Yeah.
Okay, so what we are doing is that we are irradiating now DNA. I mean, Dave told you, right? With circularly polarized light.
Yeah. So I'd like to hear one of your talks on that, Noemi.
Well, I need just to be convinced about what we see, because right now we just try to reproduce and reproduce until we are convinced. But yeah,
if you ever give a talk, please invite me. In fact, I hope you will continue to invite me to listen to your talks at your institute, because they're very interesting.
Okay, well,
I don't know if you have a web page with all of the talks uploaded.
yes, so we are going to upload, if you authorize us, your talk in our astrobiology webpage. So if you go to UC Santa Cruz Astrobiology Initiative and you go into events, then you will see all the astrobiology colloquia that were given until now.
Okay.
And you can click on them, and then you will see the recordings, because we put them on the website.
Oh, that's great. And do you have a talk loaded?
Not mine,
No?
but
Okay.
I don't have a talk yet ready on that. But
I have to invite you, to give a talk.
Okay, well, yeah, invite me.
I'll invite you at your institute.
No, but I am the host of with Artem of the astrobiology colloquium. So
Okay, that's even better, no?
We invite other people to talk. So
yeah, I saw your slide presentation you sent me, but there are a lot of details that I didn't understand from the slide presentation. So it would be really nice to hear you give that talk.
Well, if you invite me at UNAM by Zoom, I can give a talk.
Well, the trouble is that I have now retired from the UNAM.
Okay.
Because I had a bad case of autoimmune disease.
Oh yeah, you told me.
I think it was triggered by COVID vaccine. I'm not sure. I don't want to start any conspiracy theories, but it seems
my mother had an issue with vaccine as well, so I don't know.
COVID? Really? Autoimmune issue or something else?
The myocarditis.
The what? Sorry.
Well, I'll tell you.
Okay, it doesn't matter. But yeah, a lot of people seem to be.
It's like, yeah, I mean, it's a balance between how much we gain from having the vaccine save us from COVID and also the side effects, right? So
yeah. Seems to be negative. The overall result.
Maybe it depends on the age of the person as well.
Yes, of course, also.
But
Yes. But anyway, so I retired because they were forcing me to come back physically to be there. And I just couldn't even walk. I couldn't even get out of bed. In fact, it hurt so much my my joints. So then I decided, yeah, I had the years, so I decided to retire. And now I can do much more work. Because we have a lot less bureaucracy. No, it used to be 50% of my time was doing bureaucracy, bureaucratic work.
so now you are still in Mexico, right? I mean, you don't travel.
Yeah, I'm close to Mexico City, about 100 kilometers to the south. And that's important because we get a lot of sunshine. And for those who have autoimmune disease, vitamin D is most important. So I'm getting better, but I still have my hip, which is giving me a lot of pain. So there's no more cartilage. The cartilage has been destroyed, so it's just bone on bone. But I have found that there are people that have been able to reproduce the cartilage, not in the same state as it was originally, but they are able to produce new cartilage. So that's what I'm hoping for now, but it's a very, very slow process, especially at my age. So, but anyway, I keep in contact. They still have me in the department here in
Okay,
the UNAM.
so let's do this. I mean, I think we are going to do more experiments in December, and I can contact you in January and we can chat.
Sure. Okay.
I'm curious to hear about them because right
Yeah.
now I still want to perform more tests before before we are convinced, right, about what we see. So.
Yeah.
Because, I mean, if there is a difference in power between the two beams, then you want to make sure it's not that is affecting the result, right? Because it's never perfect. So.
Yeah. Well, this is still an open question. There are many theories and nobody has really proved any one of them. But well, this we have done more or less an experiment, but we haven't done the homochirality experiment. No, the denaturing is there.
Yeah.
The homochirality is not there, and I won't be doing it anymore. So maybe I can convince somebody up there to do it.
We can do it, maybe.
Yeah, that would be interesting.
We have a laser, and we can maybe have some cuvette, which would be temperature controlled, right? So we can discuss that and see if
First thing would be to try to reproduce what we have found, no? If that can be even reproduced.
with non-polarized light, and then add the polarization to see if there is an effect.
Yeah. Yeah, denaturing first and then with polarized light.
Yep.
Yeah, do you have a polarized light spectrometer?
No, what we do is that we have two beams.
Mm-hmm.
One is circular left and one is circular right.
Oh, okay. Mm-hmm.
So it's at exactly 266 nanometer.
266? 266?
Yeah, we cannot change that.
See, that's maybe also part of the problem because as I was just explaining to your colleague that, for example, you can get production of diamine dimers at 270 nanometers. But if you go to 220, then you can break those dimers.
Exactly, yeah.
So it might be interesting to do it instead of with a laser, to do it with a light source like deuterium.
Yeah.
To get the whole spectrum, which is arriving at the Earth's surface.
Yeah, for now I only have one wavelength, so I have to finish what I'm doing.
Mm-hmm.
But then yes, that's certainly possible. We have like other type of.
Yeah. You have a postdoc position now, or you are a faculty?
I'm looking for a faculty job right now, so I'm like applying everywhere again.
Okay.
But I still have money with my Sloan Matter to Life grant for until August, so I can buy stuff, like DNA or mirror even DNA because they're producing it.
Yeah, that's great that your project was
I-
funded.
So,
Yeah,
yeah,
to
so
some
I,
of
yeah,
the
yeah, yeah, I was really happy. I mean, you were like an advisor on this, right? I sent you it. So, and then
no, they didn't send me, but that's great
I
that
sent you the proposal.
Mhm. Yeah,
But then what we did is that we spent a year trying to tune the power. That was really complicated
to
because
change the power, the intensity.
to put the same intensity in the two beams, because when you buy a beam splitter,
Mhm.
like it's never perfectly 50-50, right?
Right.
So
Mhm.
it's always something like, I don't know, let's say 51-49, right? For example. And then you want to correct for this. And
Yeah.
also the laser is not stable. Sometimes, one beam is a bit more,
Right.
sometimes it's a bit less.
That's
So,
right. I think the
I had to-
theoreticians just
yeah,
don't understand how complex experiment is. It's very complicated. Our
Yeah.
experiment too, it looks simple, but it was also required a lot of work to get to
So
where we finally got.
I really spent a year trying to
Yeah, easily.
So I bought like filters, and also I bought a super complex like monitor that actually have two detectors that
Mhm.
measure simultaneously the number of pulses, so the number of photons, and
Mhm.
to get the radiation dose in the two samples as a function of time. So then we know exactly what the sample get, and we are able to say they get this amount of radiation dose up to the difference is like smaller than 1%, right? And then we would know if we observe an effect which is 10%, that it would be due to polarization, right? And not to because if no, you could say, okay, if you see a difference, maybe it's the power between the two beam. Maybe it's not because one is polarized left and one is polarized right, correct? So this is why I'm saying it took so long and we are still in the test, but we are getting result and it's always the same. But I just want to, make more and more tests. So
Yeah.
we can say like anything.
Well, my thesis experiment was in nuclear physics. It's called polarized proton-proton Bremsstrahlung, which
Yeah.
is very difficult because there's a very big background. And so we were like 5 years into my thesis project, and we still hadn't seen data, useful data. So everybody gave up, except maybe 3 of us, myself, my supervisor, and another postdoc. And it was the 3 of us who finally realized what was going on and what we needed to do, and we did it, and we finally got the results and published, and now it's been cited quite a bit. So yeah, experiment is very, very difficult.
Yeah.
Most people don't understand that.
Well, I didn't before. I thought I will buy a beam splitter. It will split the beam 50-50.
Yeah, beam split.
Yeah.
That's another route, huh?
Yeah, but we'll see. I mean, I'm just doing all this to better understand like DNA really, because like
Yeah,
at
well, the work you're doing now is going to be useful in the end for something.
and so yes, as when I get something that I'm convinced about, I will tell you and we can have a Zoom.
Yeah, great. Yeah.
And
And
you again
and put me on your list, your email list, so I get invited when you're gonna give talks.
Okay, I will.
Okay, that's great. They're always gonna be through Zoom or hybrid.
This thing no, I mean sometimes we are inviting people, but it's like very slowly going back to normal, right? Because there's still COVID, outbreak.
But
So,
it would be nice if you could keep the Zoom going also.
yes, so people can attend.
Oh, that's really great. And you're having a good time in California?
Yes. I mean,
Yeah.
I'm European at heart, but it's still, there are so many advantages in being here for research, because in France it's not doing so well right now. So
Yeah, all of Europe, I think, is in problems now with the energy problem. No?
Okay.
Mhm.
So, okay, I will have to go soon because my family will come and I have to help.
Okay, sure, Noemi. Yeah, there's no more questions, and well, we can say goodbye and thank you very much again for the invitation.
Thank you so much, Karo, for your time. Thank you for staying for additional discussion. It was a
Yeah, and
very
it's super late for you, no? Now it's like
No, it's still early.
but we will let you
Well.
enjoy your weekend and Friday evening, nevertheless. Yeah.
Yeah, well, thanks, Artem. That was really nice of you also to set me up and invite.
So when we put it on the web, we will send you it will be the same link.
Okay,
And you can go and see the previous, if you are curious, the previous.
I can't hear you anymore. I'm sorry.
Microphone is
Yeah, I'm saying we will send you the link when we will put your talk online.
Okay, yeah, great. Yeah,
Yeah,
thanks, Noemi.
access to the other speakers.
yeah, exactly.
Okay, well, so
Thank you so
bye-bye.
much, Karo.
Thank you.
Thank you very much, Artem, Noemi,
Have a-
Andres, my students also connected. So thanks to my students also for connecting.
Yeah, and we stay in touch. Okay, well, see you.
Okay, so goodbye.